Spectrophotometry Lab video by Rachel, Jaime, and Morgan. Here we are making our blank by filling our cuvette with half water and half ammonia. Here we are transferring sufficient but not excess stock solution to our cuvette. Here we are transferring our first dilution into cuvette. We made the first dilution by manipulating the M1V1 equals M2V2 equation. Here we are transferring the second dilution into cuvette, which we made by utilizing the M1V1 equals M2V2 equation. Here we are pipetting our third dilution into the cuvette. We made that dilution through manipulating the M1V1 equals M2V2 equation. Here we are properly zeroing our spectrophotometer by wiping the sides of our blank cuvette clean, placing the cuvette in the spectrophotometer with the clean sides in the beam, properly shutting the spectrophotometer, and then hitting reads and 0 on our program. Here we are using the scan program on the spectrophotometer in order to find the peak wavelength to scan all of our dilutions at. We do this by using the stock solution. It is important to scan across all wavelengths because every molecule is wavelength specific. The optimal wavelength at which the absorption could be read is called the lambda max. A broader range of wavelengths can also allow you to determine the purity of a substance. Two lambda maxes can correlate to two components of one mixture. Lambda max refers to the wavelength along the absorption spectrum where a substance has its strongest photon absorption. You can use lambda max as a parameter to compare different qualities of all types of molecules and substances. The Beer-Lambert law states that the quantity of light absorbed by a substance is equal to the molar absorption coefficient times the concentration of the substance times the path length of the light through the solution. The proportion of the light absorbed will depend on how many molecules it interacts with. A highly concentrated solution will yield a high absorbency, while a highly diluted solution will yield a low absorbency. Failing to zero at the machine with a blank will result in a higher absorption than what is expected, because instead of the absorption of water being canceled out, it is added into the absorption for the solution. Here, Rachel is properly using the spectrophotometer to find the absorbency of the stock solution. Here, Rachel is using the Simple Reads program on the spectrophotometer to find the absorbency of dilution 1. She is making sure to close the spectrophotometer before scanning, and she carefully removes the sample from the spectrophotometer and shuts the door when finished. The absorbency of dilution 1 was 0.0655. Here, Jaime is properly using the spectrophotometer to find the absorbency of dilution 2. The absorbency of dilution 2 was 0.0070. Here, Jaime is carefully removing the sample from the spectrophotometer and closing the door. Here, Morgan is properly using the spectrophotometer to find the absorbency of dilution number 3. The absorbency of dilution 3 was negative 0.0498. Here, Morgan is properly removing the cuvette from the spectrophotometer and closing it when finished. Here, Rachel is properly disposing of the materials used in the spectrophotometer room. Here, our lab notebook is clearly shown, and it includes labeled samples of decreasing concentration, absorbance data for all samples, and all significant figures are recorded to the correct decimal places. Here, Jaime is properly cleaning our materials with soap and water to be used for the next lab group. Here, Rachel is properly disposing of all the liquids used in the lab by pouring them down the sink, and then she cleans the beakers out with soap and water. Here, Morgan is drying the materials used in the lab for the next group.